Greetings. My name is Cheetah. I'm a farmer, consultant, and applied mycologist based out of Northern California. The purpose of this video today is to give you kind of a basic introduction to the concepts of mushroom cultivation. This is a follow-up to a video I did talking about the fungal bioremediation work, the mycoremediation work I've been doing over the past couple of years. People are always interested in ways of getting spores or different ways to apply fungi to the land. And it's occurred to me that a lot of folks don't have a solid concept of how fungi are cultivated. And really on the introductory level, as kids, we're taught about photosynthesis and how plants work, but we don't have a clear understanding of how fungi work. So hopefully we can run some of that down for you. So you can go back to my previous slideshow and check out kind of a more in-depth description of this, but fungi are these organisms that are heterotrophic. That means they use sources of carbon, synthesized sources of sugar synthesized by somebody else. And so they've got a couple different lifestyles to get their energy, their nutrition. Right up here, we're talking saprophytic. So saprophytic are fungi that eat other organisms that are dead. So this would be your wood decaying fungi. When you happen across a rotten log in the woods, that's a saprophytic fungus. The next kind of component of this is symbiotic fungi. So as gardeners, we're familiar with mycorrhizal fungi or possibly even endophytic fungi. These are the fungi that work in conjunction with plants to assist them in their process of photosynthesizing sugars in some way, shape, or form, or perhaps defending them from parasitic organisms. But basically, these are fungi that work in conjunction with other organisms to get their source of energy, their source of carbon. Parasitic fungi, these are fungi that attack living organisms. So you probably have some of these living in your body right now. They attack your plants in your garden in the forms of different disease and pests, basically. And so all this is just a kind of roundabout way of saying that there's many different types of fungi that get their energy in very different ways. For the purpose of today, though, we're talking about saprophytic fungi. Again, these are the fungi that eat dead things for their lifestyle. And so this is a very, very generalized life cycle of a fungus in which when we see a toadstool or a mushroom sprouting in the woods, this is just the reproductive organ of the fungus. Its sole purpose is produce progeny in the form of spores. So spores can best be equated very simplistically to like a piece of pollen. So it carries half the genetic complement. It's only half of the organism there. When it lands and finds favorable conditions, it'll germinate and it will find a compatible individual, and they will merge via these hyphae. So hyphae are these filaments. It's the growth habit of fungi. So again, can perhaps be best equated to the root of the fungus. So these very narrow single cell tubes are grown through the environment. They find a compatible individual and they merge. They exchange genetic material, and then they can grow forward in their lifespan as a complete genetic individual, as a whole self. When they've amassed enough nutrient, or perhaps they happen across hard times and feel the need to reproduce, that's when the young mushroom starts to grow. And again, we then have the mushroom, the above ground reproductive portion of the fungus. So again, this is very, very generalized. This is a super simplified way of looking at fungi. There's a million different ways to look at it. There's so many examples that I can't go into right now. So again, this is a very, very simplified perspective of fungi. So going forward with that, we're just going to jump right into basic mushroom cultivation. I'm coming at this today from the perspective of the very simplistic, low-tech methods of mushroom cultivation. My bioremediation work came out of losing my home and my farm in a forest fire, which is now becoming more and more prevalent. We're getting these urban firestorms. So upper left-hand corner here, I had a fairly nice laboratory and it was destroyed in this forest fire called the Camp Fire here in Northern California. And the fungi, while they were burned and scorched, they lived through the process. And so inspired by that, I've built up a very, very simplistic laboratory in a cargo trailer here. And so that's my mushroom farm as of now. I've got a small 7x16 space that I grow all my mushrooms out of. And so this is kind of my way of being able to get back to work and reclaim my farm, but also I've learned some tricks along the way that you don't need a super high-tech laboratory to grow mushrooms. There's some shortcuts and there's some easy ways to do things that don't require so much technology, and that's kind of what I want to share in this video. Mostly we're going to be talking about oyster mushroom, Pleurotus austratus, and others. We'll talk about specific species in a minute, but they're a super tough mushroom, they're delectable, they're medicinal, and they're kind of the flagship bioremediation fungus, and so mostly we're going to be talking about oysters today. So as a mushroom farmer, basically what we're doing is we're facilitating the life cycle of this fungus. We're creating the best possible conditions for this mushroom to grow in. And so there's a host of different ways we can go about that, but basically we're just trying to set them up for success. That's our job. So the fungi, again, we're talking about saprophytes, and so they're wood eaters. So the two primary components of wood is cellulose and lignin. So cellulose on this side here is this very simple glucose subunit linked in these very simple chains by this oxygen linkage, very linear molecule. Some fungi can just eat cellulose. And so when you happen across like a rotten log in the woods and it's like this brown rotted out log, that has been eaten by a cellulose, what we call a brown rot fungus, and has left the lignin skeleton behind. Lignin on this side here is a irregular, bony, very tough, very recalcitrant molecule. And only certain fungi can break it down. And for the most part, when it comes to edible and medicinal fungi, we're looking at these white rot, these lignin degrading fungi. And oyster mushroom is one of those. And so that's what we're talking about. So we want to make it as easy as possible for the fungi to do their job, to eat these molecules. So commercial mushroom cultivation is this slow and steady process of propagating fungal cells. Most commonly, we start with a tissue sample, a very, very small portion of fungus. This could be a liquid culture. This could be, we take a mushroom itself and we cut a piece of tissue out of it. And what we do is we take it and we put it in one of these Petri dishes. So what this is a glass plate with sterilized, basically a vegan gelatin inside of it. That is the surface upon which the fungus grows. And so you can kind of see that here. So we create this sterile, nutrient-rich media. We take a small cluster of fungal cells and we place it on this substrate. We can also store them in test tubes. And so we grow it across the surface of this plate. And then typically, our next step for production is moving them onto a grain substrate. So what we do is we take a mason jar of grain. We have to prepare it in a certain way, but we sterilize it and then take a chunk of that Petri dish and put it onto this grain. And so what that does for us is it creates a nutrient-rich pelletized material. So it's these individual units of fungus that we can then propagate onto our next material. So with our pelletized mycelium, our individual units of mycelium, we then go on to these spawn bags. So these spawn bags, what this is a polypropylene bag. So it can withstand very high temperatures and can be sterilized without melting, without falling apart. And it's outfitted with this microporous patch. So what this is a piece of plastic that will allow the passage of air, but not the passage of contaminants. So this tiny little patch will filter air but not let anything larger than 0.3 microns through. So any mold spores, any competing bacteria, anything like that cannot pass through that substrate. And so from our, what we call these, the spawn bag, we can then jump onto any host of material. We can move them onto hardwood logs. We can move them onto manure-based substrates in the case of like agaricus or others like that. Or move them onto more wood chips or straw, as is the case commonly with the oyster mushroom. So basically, what we do is a slow and steady progression from a very small cluster of cells to hundreds to thousands of pounds of myceliated substrate, what we call, again, what we call spawn. So a lot of different materials can be used. A lot of times for our applications, we use hardwood sawdust or wood chips. In the case of the oyster mushroom, that's a great choice. Composted substrates for, again, like the agaricus type mushrooms. There's a handful of others that use composted substrates, but not so much what we're talking about today. Rice straw is what I use a lot for my oyster mushroom cultivation. I'm here in Northern California where we have extensive rice fields, and so certified organic rice straw is readily available. Also cereal straws, we're talking about like wheat and oat straw, can commonly be used as well depending on its availability. So we can grow them onto hardwood logs. That's a discussion all itself. You can take little furniture dowels and grow your mushroom onto furniture dowels and drill holes and hammer them into logs. That's a common technique. But you can also use sawdust. You can cut kerfs with your chainsaw and pack them with sawdust. And there's different ways that we can get mushrooms to grow on logs, which is kind of a perennial method of growing mushrooms. And then a whole host of woody agricultural wastes, everything from cotton waste to corn stalks, corn cobs. A lot of research has been done in terms of how to convert agricultural wastes into mushrooms. Liquid cultures, I'm not going to talk a lot about right now, but that's a great way for a home cultivator to be able to propagate fungi on a micro scale without a lot of sterile equipment. If you're interested in liquid cultures, check out what they call the airport technology, which is a way that we're able to inject fungi directly in through the lid of these grain masters and doesn't require some of the specialized equipment that others need for propagation. And then there's a whole host of other methods. Cardboard, fungi for some reason having a strong affinity for cardboard. You can soak it in hot water, separate the corrugates of it, introduce the fungus in any kind of host of ways into the layers of cardboard and kind of lasagna farm it that way. And that's a great way for home gardeners to apply fungi to their garden plots. We're going to talk a little bit about burlap culture. For some reason, fungi have a strong affinity to woven burlap materials. And if you follow the link below, talk a little bit about coffee ground culture. Basically, coffee grounds are super high energy, they're super high nitrogen, and it's a fun kind of home scale way that you can get started growing mycelium. So low-tech methods, I mean, commercial farms, they use all this high-grade sterilization, these filtered air. But for the home methods that we're going to be looking at, yeah, it's really just kind of farm scale stuff. So yeah, again, soaking cardboard in hot water, separating those corrugates, introducing the fungus that way from a spawn bag, what have you. Burlap, or what they're calling bunker spawn. So coffee bags have become really popular as well. Stem butt propagation is a way in which when you harvest a mushroom from the wild, the base of that mushroom has a cluster of actively growing cells. And so when you trim off that portion of the fungus that would normally just throw in the compost pile, you can introduce that in between the layers of your cardboard substrates, get the fungus to leap off that way. And using virtually no technology at all, you can get a living growing fungus growing that way. Spore mass slurry, say you're hunting in the woods and you happen across a fungus that is a choice edible, but maybe it's been totally roached out by insects or something like that. You can take that mushroom, grind it up, put it in a 5-gallon bucket of water, maybe a pinch of molasses to help it kind of that initial leap off, stir it up and spread it on your wood chip bed or your compost bed. And it's a way that potentially doesn't always work, can propagate fungus on your property. I've had some good success with chainsaw bar oil. So bar oil is the lubricant for your chainsaw chain. So what I'll personally do is I take like a cookie sheet and I pour some bar oil into that. And again, I've got my mushroom that's either too far gone to eat, or maybe it's roached out from insect damage. I place that on a screen, like a screen door or a mesh, piece of wire mesh above that cookie sheet of bar oil. It deposits its spores into the bar oil, and then I feed that into my chainsaw. And then that way, anytime I'm cutting wood in the forest, I'm dropping a tree, what have you, I'm spraying spores throughout my property. And so I've had some good success, specifically with reishi, getting mushrooms to grow that way. And so it's kind of just use your imagination. There's a dozen different ways to propagate fungi in very low-tech methods. So methods of sterilization or pasteurization. So pressure sterilization here, these are all-American pressure cookers. So these are basically canners that would be used for food preservation, but for our applications, what we do is we take some of these spawn bags, we fill it with our substrate, our material of choice, be it hardwood sawdust or grain, what have you, and we put into these pressure canners and run them at like 15 psi, for a couple hours, depending on what you're doing. And that's kind of like the high quality sterilization. It kills everything. You've come out with a really clean material, no competing mold or fungi, no competing bacterium. Pasteurization is more the farm scale or thing I kind of lean towards when it comes to beginning growers. So in these 50-gallon drums, we'll stuff these with our straw of choice. I use certified organic rice straw, fill it with water, and then we put a burner under there, a propane burner, so something typically like used for home brewing. 80,000 BTU burner is probably sufficient. And in this 50-gallon drum, we heat it. I like to reach 180 degrees, and then we drain off that water after a period of about 2 hours. And what that's functionally done is, again, killed that competing mold, that competing bacteria, anything that's going to want to fight for the nutrients that your fungus is looking for. And for our applications, we just weld little taps, little valves into the bottom of them. So for our applications, we can bring this initial barrel up to temperature, drain the water off, and just using 5-gallon buckets, transfer it over so we can just squeeze a little bit more sterilization out of that same energy that we've dumped into it. Basically, we've brought that water already to 180 degrees, and we transfer it to our second barrel, and then that way we don't have to do the whole process over. Don't do it more than twice, probably. After that, there gets to be too much of accumulation of nutrients, and you're going to see poor mycelial growth. It's just going to be too hot. Something that we've been latching onto more, doing more frequently, is the lime pasteurization technique, and I'm going to talk about that a little bit more in a second. Basically, it's a low temperature chemical sterilization of the substrate. There's other ways you can do this with hydrogen peroxide. There's a twofold effect to hydrogen peroxide. Basically, you have the initial oxidative effect of it bursting cells and attacking existing mold and fungi in the material. But then hydrogen peroxide, that's H2O2, it'll convert to water in the end after it reacts. And then also you have the added effect that many of the enzymes used are produced by fungi are peroxide assisted or peroxide activated. And so this is kind of a unique way that hydrogen peroxide could be used in sterilization, or at least cold water pasteurization. There's lots of other methods. Bleach, not permitted in organics, but it'll sterilize a material. Soap, wood ash works very much like lime pasteurization. Cold water fermentation is where you soak your material for a little over a week. The whole system goes anaerobic, that is, there is no oxygen present, so it kills off all the oxygen breathing organisms. You drain off the water, and then you have a window of opportunity that your oxygen breathing fungus can leap onto the material. But it's a very stinky method of cultivation. So I want to talk about lime for a second. There's a lot of different things in the agricultural sector that we call lime. There's, basic oyster shell that we use, that's calcium carbonate as a way to adjust pH in our soils. We have dolomitic limestone, which is, has a high magnesium component. When we talk about lime pasteurization in mushroom cultivation, we are talking about hydrated lime. And even there, we have to parse out what we're talking about when it comes to hydrated lime. So some people talk about using this type S hydrated lime for this lime pasteurization method, and that's not quite right. That stuff has a little too much magnesium in it. What you want to do is try and find a food grade high calcium hydrated lime. So here you can see we're looking at over 70% calcium content and a 1% magnesium content. The math is a little funny on this sack, but this stuff has been working great for me. It's definitely more expensive than the type S hydrated lime that's used for masonry. You can get away with it. I've used it before and had success. But again, the purpose of this material is you mix it with water and your substrate. It raises the pH so high that no competing organisms can persist in the environment. You soak it in the solution of calcium hydroxide for 16 to 24 hours, probably not more than that. Then you drain off that solution, and then this lime, this calcium content, becomes a nutrient source for your living growing fungus. So for our applications, when we're doing these 50-gallon drums, what we'll do is we have our 5-gallon bucket on the side. We take about 300 to 400 grams of calcium hydroxide. That's a little over 3 cups. And we mix it in our little 5-gallon bucket, make sure it's stirred up really well. We fill our 50-gallon drum halfway up with straw, dump half that 5-gallon bucket in there, 50% of that 300 grams into the bottom half of the drum, start filling it with water, add more straw, stir up our 5-gallon bucket again, dump it in there. And so now we have the entirety of our 300 grams of calcium hydroxide fairly evenly mixed into this 50-gallon drum. We do the same thing with our other 50-gallon drum because we typically run 2 batches in one day, usually more if we can. And it sits there, again, 16 to 18 hours, and then drain it off, and then we proceed with our inoculation process. And again, there's a link in the description below for the next video, which shows a little bit more hands-on how you can kind of execute this. So in terms of applying fungi to the land and what I've been doing over the last couple of years with bioremediation, the 2 primary materials I've been using is this straw, spawn bags, which again, please check out the other videos because it'll make it a lot more clear. And then also this jute. We talked about burlap. And so here in this livestock trough, I've soaked my jute material for 18 to 24 hours in a calcium hydroxide solution. And then I inoculate it with the fungus. And what it produces is this burlap material that is fully myceliated. It's got a living growing fungus on it. And I can just roll it out into the world and have an immediately active mycelial mat. So here's kind of a little bit more on that. Here I have my burlap roll. It's been sterilized. I've got my mycelium growing on, in this circumstance, it's rice hulls. And so I spread my mushroom rice hulls over the surface of this sterilized jute material, roll it into this, this reel here, and then I stuff it into a garbage bag for maybe 2 weeks it takes to colonize. And then here you can see a fully colonized jute roll, and it just started to sprout a fungus here. And so it's a great material because for bioremediation, especially because it's nutrient rich enough to support the growth of the fungus, but not so nutrient rich that when we put it out into the world, the fungus is just going to stick to its substrate. It's going to want to grow out beyond this and search out additional nutrient, which is kind of critical for our bioremediation practices. When it comes to fruiting out fungi, they're going to need a handful of things. Humidity, 70 to 90%, kind of depends on what species you're growing, but that's kind of a good rule of thumb. Good humid environment. Air, mushrooms breathe oxygen, so a lot of times when we put our mushroom bags out into the grow house, we like to just open up a couple larger holes in them, so that breath of fresh air will stimulate fruit body growth. Light, it's a misnomer that mushrooms grow in caves. They do need some light to grow. Here, this is the coral form of a tiger sawgill fungus, where it's creating this branching structure, just reaching out, trying to find light. So indeed, they need a certain amount of light, maybe not too much during the summer months, but some is required. And then temperature just kind of depends. I mean, here we have the pink oyster. This is a tropical mushroom. It likes 90 degrees, 100-degree temperatures versus, say, maybe our blue oyster, which is more in the 50-degree temperature range. So oyster mushroom, again, that's the primary mushroom we're talking about. There's a lot of different things called oyster mushroom. I mean, even just with Pleurotus austratus, there's the pearl oyster, there's the dove oyster, there's the elm oyster. So I like to refer to it as a species complex. I think that's a little bit more accurate, where you get some blending between species. Here we have the pink oyster. Even then, you get a lot of different names for it. Pleurotus sage-orchajor, Selmeo selmestris, Deja Moore. So this is a pan-tropical mushroom. It's found everywhere from Florida to Indonesia. And it's, again, that high temperature strain. Here we have the golden oyster. This is Russo-Siberian species, so native to like Russia, basically. And great wide temperature strain. It can tolerate some of the higher temperatures. It'll drift into some of the lower temperatures. So a lot of different things that are called oyster mushroom, including which the phoenix oyster. So I like to put this into a different group from kind of the main oyster complex, because the interesting thing about Pulmonarius is that it can tolerate conifers and hardwoods. So when I say conifers, I'm talking about pines and things like that. So if you're in a region that's poor in oak or poor in rice straw or something like that, you might want to turn to the phoenix oyster as your choice of mushroom. It's more limited, at least the research indicates it's more limited in its bioremediation potential in terms of its heavy metal uptake, but still a great aggressive fungus. So again, this is just talking about oyster mushrooms. This is the wide world of fungi, and there's so many more things that can be said, but this is hopefully helps you to understand some of the growth habits and ways that you might be able to start doing some small scale mushroom cultivation for yourself, or some methods for fungal expansion for your own bioremediation project. I've got a couple more videos. Again, links are in the notes below. My contact is down there as well. So if there's any way I can help, please do let me know. Thank you very much, and may the spores be with you.